A very good morning to everyone. Welcome to the 2021, the Advancement of Arts and Technology for Humanity, the third annual Symposium of Arts, Technology and Humanities 2021. Once again, good morning, everyone, and welcome to the second day of the conference. Yesterday we had the ICPA, and now we have this creative arts. Well, I would like to extend my gratitude for all of you for already attending this opening ceremony, and Honorable Rector of Institut Seni Indonesia Yogyakarta and the Vice Rectors and all the higher officers at Institut Seni Indonesia Yogyakarta. And also good morning to all respective partners of university and also presenters, and especially good morning to the moderators of today's event. Well, before we start today's conference, I would like to walk you through the agenda for today. After the opening, we will go straight to the parallel session, which is actually divided into sessions, but we will put it at once after the keynote presentation. And after the parallel session, we will go back to the keynote session, which is also forwarding us to the closing of this conference. Well, ladies and gentlemen, before we start, let us honor and listen to the Indonesian national anthem, Indonesia Raya. Thank you. So that's the Indonesia Raya for Indonesian National Anthem. Ladies and gentlemen, and the next agenda will be welcoming speech from the General Chairman of Creative Arts and also the ICPA. To Dr. Samuel Gandang, time is yours.
Distinguished guests, respected speaker, invited presenter, and participant of the annual Symposium of Art, Technology, and Humanities 2021, ladies and gentlemen, a very good morning. It is an honor for Institut Seni Indonesia Yogyakarta to organize the annual Symposium of Art, Technology, and Humanities 2021 with two conferences held this year. This year, we are proud to hold ICPA International Conference on Performing Arts and Creative Arts, International Conference on Intermedia Arts and Creative Technology 2021. We would like to share our deepest appreciation to welcome all respected keynote speakers from various countries and participants of the symposium in this special occasion. We are very grateful and these two conferences can be held this year, even though it should be held virtually. but we are able to contribute to these two conferences and to share best practice and knowledge with others. This event is indeed being indispensable for scholar, professional, and academician to meet and exchange ideas on their multidisciplinary interests related to arts, technology, and humanities, and particularly to discuss the current global challenge during the COVID pandemic. Holding up two conferences in this symposium, ICPA and Creative Arts is aimed at facilitating the science forum with representation of valuable response and commitment to expand new scientific principle, policy, and application in this pandemic issue. The topic brought by this two conference are means of conveying to the global academic sector in which art is emerged as an expression of millennial era artists by utilizing the existence of technology and the presence of a digital world that could present the actualization of works, rise the process of art creation in the digital domain. Technology, humanity, arts, and music become the media of contact since they are full of creative potential and social solidarity and cohesion, which become the source of information and insight in relation to social issues in a digital world, as well as their discussion. Ladies and gentlemen, in responding to the discussion of the issue, we are privileged to have among us experts from several universities of our overseas partner, Hungarian University of Fine Art, Hungary, Academy of Fine Art, Vienna, Austria, Tasmania University, Australia, University of Bristol, United Kingdom, Tainan National University of the Arts, Taiwan, DPFA Academic World and Health, Germany, and Mai Fah Luang University, Thailand. In addition, there are also official local partners for this symposium, such as Universitas Sanata Dharma, Universitas Islam Indonesia, Universitas Kristen Duta Wacana, EDS Lab, and Mustafa Bragama University, which have already been contributing to succeed this academic forum. On behalf of the Committee of Annual Symposium of Arts, Technology, and Humanities 2021, we would like to express our gratitude to all keynote speakers, Professor M. Tui Mariantha, PhD, Professor Dr. Iswan Eros, Dr. Svenja J. Karatz, Dr. Stephanie Wusit, Professor Dr. Johan, Professor Michael Ellison, Professor Madam Mantelhood, Professor Marcus Tweck, and Professor Pakarawat Siti Prapapon. All respected participants from various universities and institutions in Indonesia and several countries, outstanding reviewer of both conferences, and all parties who have demonstrated the commitment to make the symposium successful. Last but not least, I would like to send a very good appreciation for Rector of Institut Seni Indonesia Yogyakarta, Vice Rector for Academic Affairs, and the entire academic members of ISI Yogyakarta, who have given us the opportunity and support to hold this symposium with two international conferences, which shortly will be taken place for 2 days. Ladies and gentlemen, we strongly expected this academic forum will fruitfully produce important outcomes to develop strategic way and real solution in facing up the current global challenge in this pandemic situation through the outstanding academic response. We wish you all fruitful and productive discussion. Thank you very much.
Thank you, Dr. Samuel Gandang, for the welcoming speech. Well, honorable guests, ladies and gentlemen, now it is time for us to listen to the opening speech from the Rector of Institut Seni Indonesia Yogyakarta, Professor Dr. M. Agus Burhan, M.Hum.
Excellencies, distinguished guests, speaker and participant of ICPA and Creative Art 2021, ladies and gentlemen, I am really privileged to have the pleasant task of welcoming this distinguished gathering. It is my honor to have all of you in this academic forum that is indeed an indispensable activity for all art scholars. Indonesian Institute of the Art Yogyakarta, or ISI Yogyakarta, would like to extend deepest appreciation to welcome all participants of ICPA, or International Conference of Performing Art and Creative Art, or International Conference on Intermedia Art and Creative Technology 2021. As ISI Yogyakarta has hosted the previous seminar, this year we are honored to present the annual Symposium of Art, Technology and Humanities 2021. Even though it should be held virtually, but are very grateful that these two conferences can be held. Ladies and gentlemen, we all experience an unprecedented situations with the global COVID-19 pandemic and cause new life changing. The influence massively happen in our daily life. Its organs and adoption in all life sector, including in the art, technology, and humanities. The topic brought by these two conferences are names of conveying of the global academic sector that art is emerged as an expression of millennial era artists by utilizing the existence of technology and the presence of digital work by corporations, the actualizations of works, raises the process of art creations in the digital domain. The development of the art creations and the realizations of intermedia art enrich the definitions of art that manifests itself in the digital world and gradually increases the impact on humanities. Ladies and gentlemen, on behalf of Institut Seni Indonesia Yogyakarta, I would like to express our sincere gratitude to all keynote speakers of these two conferences. Professor Tui Mariantha, PhD, Professor Dr. Isvan Eros, Dr. Serenia Karatz, Dr. Stephanie Woodsist, Professor Dr. Johan, Professor Michael Ellison, Professor Made Madelhood, Professor Marcus Stoex, Professor Pakarwat, Siti Praparon, and all co-hosts University Islam Indonesia, University Sanata Dharma, University Kristen Duta Wacana, University Mustafa Beragama, IDS Lab, Hungarian University of Fine Art Hungary, Academy of Fine Art Vienna, Austria, Tasmania University, Australia, University of Bristol, United Kingdom, Tainan National University of the Art, Taiwan, GPFA Academic World of Health, Germany, Mai Fah Luang University, Thailand. Staff and faculty members of ISI Yogyakarta and the committee to have actively contributed the knowledge, skill, and commitment to this program. Hopefully, the outcomes of this program will be beneficial for all parties and strengthen the mutual relationship year to come. Finally, I sincerely hope You will enjoy today and the next 2 days of discussing, sharing, and networking. Thank you for your participation.
Thank you very much for the opening speech from the Rector of Institut Seni Indonesia, Yogyakarta. Well, ladies and gentlemen, honorable guests, before we go to the main session, which is the keynote presentation, I would like to extend my gratitude to Professor Dr. Tui Mariantha and also Dr. Serenia Karatz, who are already here in the Zoom with us, and also the moderator, Kurniawan Adi Saputro, PhD. Welcome. Well, ladies and gentlemen, this is the end of the opening ceremony for the Conference of International Intermedia Arts and Creative Technology, or Creativearts. And now we are heading to the main session, which is the keynote presentation for the session 1. In session 1, we will have two keynote presenters who are Professor Dr. Tui Mariantha, MFA, PhD, and Dr. Serenia Karatz. And this event will be moderated by Kurniawan Adi Saputro. And before we start, I would like to introduce you to Kurniawan Adi Saputro. He is the lecturer of Photography Department at the Faculty of Recorded Media Arts, Institut Seni Indonesia, Yogyakarta, and he obtained his doctorate degree from the Sheffield Hallam University. And he's been publishing a lot of articles and as well books, and he is very active and devoted his life to research. Well, I'm sorry I cannot read all of them because that will be too long. So, ladies and gentlemen, please welcome Kurniawan Adi Saputro, PhD.
Thank you. Good day, everyone. We are in various time zones and various circumstances at the moment, but I hope that we do well and we are blessed with health in this time of pandemic. Our first conference was in 2019, and this year we are holding our second International Conference on Intermedia Arts and Creative Technology, or abbreviated as Creativearts. This year, it is focusing on the theme of the advancements of arts and technology for humanity. There is no better time than now to reflect on the relevance of our beloved preoccupations, namely art and technology, since in these uncertain times, we are forced to ask ourselves how we are going to continue doing what we are doing and what is the use of it. In our first session, we are joined with two distinguished keynote speakers, Professor Dr. Tui Mariantha and Dr. Serenia Karatz. Our first keynote speaker, Professor Dr. Tui Mariantha, was born in Jakarta, Indonesia, 1956. He is a lecturer at the Indonesian Institute of the Arts, Yogyakarta, and since 2013, he serves as the chief editor of the Journal of Contemporary Indonesian Art. In addition to his extensive knowledge and experience in curating and writing on fine arts, professionally and academically, he has been interested in eco-art, at least since 2014. And he has been writing and conducting workshops in this field of research ever since. Some examples of these are giving workshop on eco-art at the School of Fine Arts at the Indonesian Institute of the Arts, Yogyakarta in 2014, giving lecture on eco-art and environmental arts at Nakhon Si Thammarat College of Fine Arts, Thailand in 2019. He gave speech titled Eco-Art Through Various Efforts and Ways at Sustainable Culture Conference at the Institute of Design Bali in 2020, and doing research on the biodynamic agriculture activities of Yos Suprapto and his contribution to eco-art. which we are going to hear about today. We will have 30 minutes of presentation from Professor Dr. Tui Mariantha and 15 minutes of question and answer. Before we start, if you have any questions or response to the presentation, please write in the chat box so that the speaker can answer them in a timely manner. Without further ado, Professor Dr. Tui Mariantha, the time is yours.
Ladies and gentlemen, here is my presentation entitled Innovations of Biosynthesis in Enhancing Rudolf Steiner's Biodynamic Agriculture Teaching. Since 1968, farmers in Indonesia have continuously used chemical fertilizers and pesticides for growing rice paddy. Such synthetic substances began when the new order government modernized national farming with modern agricultural technology. World agricultural reforms of the 1960s became the legends of that era called the Green Revolution. After adopting such technology by law, the government instructed farmers to use synthetic substances for national farming. Modern agriculture system accordingly use synthetic fertilizers for inorganic seeds. On the other hand, it removed endemic paddy seeds that could not stand with the synthetic. Critical voices criticizing chemical fertilizers were far from overheard, whereas there had been so many research findings stating that the continuous use of chemical fertilizers and pesticides damaging soil structure, polluting the environment, threatening the health of farmers and the users of respective products. This report was about a book by some agriculture professors voicing a critical review on synthetic fertilizers published in 2012. However, people didn't care about this ongoing problem. Synthetic seeds, fertilizers, and pesticides are much more accessible than organic ones. Impact of green revolutions on rice cultivation practices and production system. This study showed that local farmers had stopped their traditional ways of planting. Synthetic varieties had replaced most local paddy seeds. Farmers became dependent on inorganic fertilizers and seeds. Moreover, the engineered varieties had also more vulnerable to diseases and pests, such as brown plant hopper. The circumstances described above did not work for Eusebius, an artist who questioned the conditions to articulate through his art. His acrylic paintings, entitled Waiting for Them Awake, shows his reflections on farmers' life in his home country, Indonesia. Suprapto made this work when he still lived in Australia from 1974 to 2000. At the time, he was seriously learning the biodynamic agriculture initiated by Rudolf Steiner in 1924. Steiner's biodynamic agriculture system is a farming method that utilizes bioenergy and cosmic energy to generate life force in plants to nurture seeds and produce good quality food. The system regards soil, land, plants, livestock, or farmers of a farm wholly as a living system. They are interconnected, complementary of each other. None of them is separated. The BDA system rejects any usage of chemical or synthetic substances for farming. This study aims to find out why he was so eager about developing BDA in Indonesia, How did he transmediate the BDA system initially created for subtropical climates in tropical Indonesia? What was his contributions to enhancing the BDA system? To answer those questions, the author used Arnold Berlian's theory of aesthetic engagement and George Gadamer's hermeneutic theory to understand the BDA activities of Suprapto. Concerning conceptual art, he embodied four as a series of interconnected activities that requires creativity and commitment. To collect the data and to understand Suprapto's achievement, the author used in-depth and participant observation methods and literature studies. All of these methods and the data were in use to recognize the innovations of Suprapto in pioneering stainless BDA system in Indonesia. Data and clues found. BDA uses cow manure, odor of rose quartz, herbs, flowers, plant barks to make particular compost. The cow manure is essential as material of the compost ingredients to make bioenhancers or prime starter of biodynamic. The BDA system performs a set of preparations before the planting step called the 9-step preparation to condition the soil and plants. This preparation is encoded with BD 500, BD 501, BD 502 to BD 508. The first experiments in Bangorejo village in Banyuwangi, East Java, 2009. Supraptos pioneered planting local paddy seeds by Steiner's BDA systems after 9 years of returning to Indonesia. BDA has well developed in more than 55 countries. He did not use any synthetic substances for farming at all. When studying fine arts at the Academy of Arts Yogyakarta in 1971 to 72, he met Afandi, one of the founding fathers of the modern art of Indonesia. Afandi told him that artists should not only be stretched on the subjects of art and aesthetic, but must also explore ideas, issues, and reality outside the art world. Suprapto's initiative of pioneering BDA in Indonesia, he thought of as conceptual art, connected to Afandi's views on art praxis that grew in Suprapto's art thinking. In 2015, Suprapto moved to the city of Yogyakarta, where he studied fine arts. In 2016, he managed to transfer BDA through his interactions with artists, social activists, intellectuals, and ordinary people who have been concerned with the future of the country's domestic agriculture. Suprapto thought BD 500 compost making, putting cow manure in cow horns and burying it for 3 to 4 months. Suprapto has also taught sensitivity to take advantage of local plants and herbals to make compost. Here he was showing the latang plants people usually regard it as a wild plant. It would have been costly if BDA practitioners had to buy the original materials from overseas. Suprapto managed substituting the original as in the classical formula with the endemic. Here, as an example, he substituted rose quartz powder, with the steric benzoin resins easily found in the country. Here is steric benzoin or Boswellia sacra resin. Suprapto's training in appreciating and utilizing local plants for good has indeed opened up people's ecological awareness. Keep natural environment and its ecosystem sustainability. The substitution was actually practically helpful, saving money and time. This was when Suprapto just harvested a biodynamic compost BD 500 on 18 September 2020 after 4 months buried. Made in one workshop at Bantarjo Village, Kulon Progo, Yogyakarta. Here, Suprapto helping somebody creating a vortex in dissolving biodynamic compost with water to sprinkle on the planting land. Through farming, Suprapto has practiced his conceptual art to represent his aesthetic and social engagement. Suprapto was teaching how to sprinkle the solutions of BD 500 at the right time between 3 o'clock p.m. and before 6 o'clock p.m. He also innovatively utilized the Javanese lunar calendar called Primbon to find suitable days and times to sprinkle out biodynamic solutions for soil fertilizers and nutrition for plants. Here is Suprapto's table of PDA compost making in its benefits. Quite detailed, and this is very helpful for anybody who wants to learn biodynamic. Through the work he calls in the praxis of the conceptual art, Suprapto has produced outstanding creative work in the intangible forms of a sustainable natural agricultural system, whose aesthetic value lies in its functional usefulness and ecological value. Conclusion. Suprapto's biodynamic agriculture startup was a realization for his long concern with the dark legacy of the Green Revolution in Indonesia. This concern had conceptually begun since the mid-1990s. However, it was not easy to introduce the BDA system to farmers already accustomed to synthetic fertilizers. Local farmers firmly rejected and stigmatized the BDA unclean and even superstitious. Efforts to implement the new BDA system were successful in 2016 in Yogyakarta, where he introduced the system not only to the farmers, but also to artists, intellectuals, and eco-activists. In the context of food supply, Suprapto has proven that the BDA system has adopted locally can grow local rice seeds that have previously disappeared from circulation. The production process itself is ecological and much more economical. This pioneering in farming by the BDA system in Indonesia was really original. Suprapto substituting the original herbs of standard BDA systems with endemic local ingredients was his novel achievement. Thank you very much for your attention. Terima kasih. Salam sehat.
Dear moderator and distinguished guests and also dear participant, I am going to apologize to you all because after recording my PowerPoint presentation, I feel that if I don't expose Suprapto's conventional art, I feel very incomplete. So I would like to expose as well the some works of Suprapto's works in a sharing screen. Technician, I would like to help you. Okay, this one. Yes. Okay. Ladies and gentlemen, in my earlier presentation, mostly I exposed his unconventional artwork. Here I'm presenting his conventional art that were exhibited in his solo exhibition in 2016. Sorry, 2017. This is okay. Okay. This is his work entitled "Backflow in the Eastern Horizon." This also included in the solo exhibition in 2017, entitled "High State." Also this one in the exhibition as well. Many of his works about natural landscape, about the life of farmers, and mostly about the life of local people or in daily realities of life. Some people regarded Joseph Rapto as a social realist artist, because he always exposed the reality of local people, farmers, and now in the last 5 years, he was so busy, occupied with his farming activities that he regarded his activities as conceptual art. And this is very, very interesting. And to tell the truth, I feel that, and I have observed that his social engagement arts is much more interesting than his conceptual art. But this is up to you, dear participants. I'd be happy to answer your question later. Thank you, dear moderator.
Thank you. Thank you very much, Professor Dr. Tui Mariantha, for your presentation. Now I invite questions and responses from the audience. You can write in the chat box so that the speaker can answer those questions. Before we answer questions and responses from the audience, I would like to ask Professor Dr. Tui Mariantha to elaborate more on the motivation. What drives the artists to experiment and also to be very much engaged in those years with agricultural issues? Could you please tell us more about that?
Thank you for asking this question. Actually, Joseph Rapto lived in Australia since 1974 until 2000. And in 1994, he was learning biodynamic agriculture system, and at the time he often wrote some articles about farming realities in Indonesia and also about biodynamic farming, and then sent to his friend in Indonesia to implement what I think the best for Indonesia. Actually, Suprapto was very concerned about the usage of synthetic fertilizers that have been used since 1968, and the using of the synthetic fertilizer and also chemical pesticide have been go on and on until now. Whereas many report in journals stating that the using of synthetic fertilizers and pesticides was very dangerous, not very good for not only for the environment but also for the structure of the soil itself and also for the living creatures, either the farmers and also the users of the respective product. That's why. And Suprapto tried experimenting, planting the paddy and what we call kedelai, I don't know, bean.
Soybean.
Okay, bean, soybean in Banyuwangi, but you know that the local people that had been so used using the synthetic fertilizer rejected at all and even drove him out from Banyuwangi. And the idea that he has to introduce the non-chemical fertilizers very strong. So he moved to Jakarta, and luckily he not only met with farmers, but he met intellectuals, artists, and also some people who were interested in introducing organic fertilizer and also local paddies. That's the drive that's make him so eager to introduce and also in developing his system that he thinks the best for Indonesia. That's the my addition as replying your question.
Thank you. Very interesting, Professor Tui Mariantha. Now we again, I invite you to submit your questions in the chat box and also your responses. Now we have our first question coming from Cahyono Priyanto. He wrote, some of Suprapto's painting seems to be influenced by a sense of sensuality. For example, is the high note. Do you find more works of him influenced by this this sense of sensuality?
Yes, in a sense, yes. As a matter of fact, in his exhibition in Australia, one of his work shown in the gallery being banned by the committee because he exposed nudity in his painting. So the painting was refused to be shown in the gallery, and it caused controversy about the exhibition, and also in some of his work, in his painting especially. He also often represents sensuality or nudeness in nudity in his work, but this is in context to exaggerate his idea about the powerful people that seeing that they according to Yesuprato, powerful people seeing the power as something sensual as well.
Thank you.
It's, I am so. Thank you.
Thank you, Professor Tui Mariantha. Again, I invite you to submit your questions in the chat box and also your responses. And also, Dr. Serenia Karatz, welcome to our conference. Feel free to give your comment or to join in our discussion if you feel like so.
Well, I'd just like to say thank you very much to Professor Martinez Tui Mariantha, because I wasn't familiar with Yesuprato's practice. So it's been really interesting to gain insight into the sort of innovative ecological agricultural practices, but also how that has translated into the development of sort of really socially engaged painting. So thank you very much for that.
Yeah, Professor Tui Mariantha, I have to admit that I'm not familiar enough with Yesuprato's artwork as well. So I would want you to explain to me and also to us about the relationship between the artworks and his agricultural practices. You have touched on these issues and give us example, but this is, I think it's very interesting. Today it's very common for new artists to blur artistic boundaries, but this is the pioneer, I would say, in our country. So his view of such issue, I think, is very worthwhile listening to. Please, could you tell us more about that?
Thank you for asking this question. Suprapto learned fine art in Jakarta in 1971 until 1972. And he happened to meet a big artist named Afandi, who at the time at the meeting told him that artists should not only explore ideas, creative ideas within and around the art itself. Instead, artists should also go beyond the world of art, exploring anything, any probabilities. And the idea from Afandi actually grown up in his mind. And actually, Yesuprato used to be an active member of Forest Watch, Indonesian Forest Watch, when he was in Australia. So he can see that the relation about creativity, strategy with art similar. Because in art, people expect to be creative. If you be creative, it means that you have to present novelty or originality. You have also to present effectiveness, and also you have to bring innovations that have value. In farming, especially in strategic farming, farmers or activists also need creativity. Either art or farming or whatever it is, when they need creativity, they have to think about strategy, effectiveness, innovation, novelty, and also commitment. And in some interviews with him, I found out that the concept of art he believed that the aesthetic of art, especially the aesthetic of social engagement art, not only based or relies on the aesthetic form or artistic lines or color, but the aesthetic based on the usefulness and answering the people's problem. That I think this is a very, very good connection. And also, I would like to give more answer to you that I mentioned in my paper that I use the theory of Arnold Berliand about aesthetic engagement. In that theory, Berliand states that artists should only reflect or contemplate about problem, but the artist should also participate with the people. So his work, his activity, be part of the problem solving itself. That's what I learned from Yesus Prapto. Thank you for questioning.
Thank you very much. That's very interesting to hear, and things to be reflected on more, I think, about how artists engage in real life and social problems. Although I think it's now more common to see with the younger artists, and also easier to be said, but I think it's very hard to do. I would like to invite more questions. Another question from Cahyono Priyanto for Professor Dr. Tui Mariantha. Do you think Mr. Suprapto's work are influenced by other major painters, such as Pablo Picasso or big names in fine arts? What do you think about this?
After conducting this research about 6 months, he never mentioned that he has a role artist. Instead, he mentions much more about his experience, his interest in going to the Baduy communities in Banten. He is more interested in explaining that he exchanged experiences and knowledge on biodynamic farming to the heads of the Baduy communities in Banten, it used to be in East West Java. So I never heard that he said that he likes Picasso or others. No, but more interested in explaining, telling stories. And actually now Suprapto is in one of the villages in Banten now, being with the people of Baduy. That's what I know.
Yeah, thank you. I think people are still warming up with our session, because Yogyakarta has been raining for close to 24 hours. We hardly see sun outside, and it's quite cold for us. I think that's affecting our discussion today. And perhaps it is wise to just continue with our second presentation, so that we have more time for question and answers, and so people can go back to Professor Dr. Tui Mariantha later. Our second presentation, in my view, is intimately connected with the first presentation, in that it questions our conceptual boundaries. What is art? What is nature? What does making art mean in our time? Our second keynote speaker is Dr. Serenia Karatz. She is an Australian practice-led researcher with a strong track record of major non-traditional research outputs, operating in the nexus of art, design, science, and technology. She holds a PhD in biotechnology and contemporary art, both, from Queensland University of Technology, and she has expertise in cell and tissue culture and interdisciplinary arts practice. From 2008 until 2012, she worked as an artist in residence at the QUT's Institute of Health and Biomedical Innovation. During this time, she produced 3 major bodies of work that map her engagement with contemporary biotechnologies, including primary culture of human and fetal calf cells, tissue, and genetic engineering. Since 2013, she has worked with researchers and creative practitioners across music, design, architecture, electrical engineering, molecular biology, and biomanufacturing, and produce a range of works that explore the complexity of living systems and highlight the philosophical and ethical implications of engineered life. Today, she will be presenting her paper titled Art plus Science plus Technology, Mobilizing Post-Human Connections and Rethinking Boundaries of Self. Without further ado, please, Dr. Serenia Karatz, the time is yours.
So thank you so much, Kurniawan Saputra. Really appreciate the kind introduction. I'm just going to take a moment to swap over for my presentation. Are you able to see my presentation?
Yes, but it's on, yes, okay, it's full screen now.
Oh, excellent. So before I begin, I would also just like to take a moment to acknowledge the traditional owners of the land from which I am presenting today, the Muanina and Palawa peoples of Nipaluna, Hobart, and Lutruwita, Tasmania, and I'd like to pay my respects to their elders, past, present, and emerging. I would also like to thank conference organisers, Bang Bang Pramo, Ruby Sofyan, and Donna Hapsari for the kind invitation to present at the Creativearts International Conference on Intermedia Arts and Creative Technology. It is a real privilege to be here today and to be one of the keynote speakers. So in line with the conference theme, the advancement of arts and technology for humanity, my presentation today focuses on the intersection of art, science, and technology, and particularly the value of creative arts practice as a tool for critical reflection and dialogue, as well as revealing scientific lab practices beyond science communication alone, and furthermore, imagining the potentials of new and emerging technologies. Since I'm foremost a creative practitioner, I will use my own practice today as an example of artworks that operate across fine art, digital media, and biotechnology, emphasizing how bioarts practice can help rethink particularly Western conceptions of the human in the 21st century. So in particular, my presentation aims to highlight how art science practice can really mobilize philosophical post-human thought, which foregrounds cross-species, nature, culture, and matter entanglements, And as philosopher and post-human scholar Francesca Ferrando points out, quote, destabilizes the limits and symbolic borders posed by the notion of the human, drawing into question perceived binaries between what is regarded as human and non-human, natural and artificial, and further undermining the once really clear boundaries between life and death. So the works that I'll be discussing today also highlight notions of becoming, which is of course also aligned to new materialist and post-human thought, in that it signals a move beyond the kind of stable and unitary associations associated with ideas of being and also ideas of fundamental essence, to instead recognize that we are in fact part of a really complex, interconnected process of continual transformation. So I'll begin my presentation today with a short introduction, just to provide some context for everyone for my practice more broadly, but also to give some insight into the scientific processes that I use in the development of my work, which, as you've heard, primarily centers on the area of cell and tissue culture, but also genetic engineering. So my initial artistic background was in painting and mixed media, but following my undergraduate degree, I really transitioned into new media arts production with an emphasis on interactive video installation and also online hypermedia projects. But as you can likely tell from the examples of some of my early work on the screen here, I've always had a really strong interest in the human body, and particularly how new technologies, especially in the biomedical terrain, impact on conceptions of self and other. So during my PhD study, I was really fortunate to have the opportunity to follow up on this interest in life science and work across art and biotechnology at the Queensland University of Technology. And during this time, I became actively integrated as an artist into the Tissue Repair and Regeneration Group, which was a research group and team that focused on wound healing with expertise in cell and tissue culture. And as part of that engagement, I was trained by fellow team members, and I learned how to culture different cell types in the lab environment. So it really allowed me to be much more active and have more agency within the scientific domain. So on this slide here, you can see the first flask of cells that I received, and they were, as you'll see labeled there, SAUS2 cells. And that title references their origin as sarcoma osteogenic, which is essentially a bone cancer cell line, which was established in the 1970s from an 11-year-old girl I named Alice. So these cells, as you can see below there, are actually commercially available as a biological research product from the American Type Culture Collection, or ATCC, which is a global repository for cell lines. So many cell lines, including Alice's SAUS2 cells, as again indicated on the slide, are grown in a single layer on the base of a tissue culture flask within a liquid nutrient medium. And they must also be kept sterile in an incubator with settings that replicate the conditions of the human body. So cell lines like the SAUS2 cells of Alice are often described as immortal because, unlike healthy cells, so cells that might be taken from anyone's body, they have an inbuilt mechanism to prohibit that, and that healthy cells, have an inbuilt mechanism to prohibit ongoing replication and thus avoid errors creeping in, which is called the Hayflick limit. Cell lines can continue to divide almost indefinitely. So as such, they can continue to grow and be split into more and more tissue culture flasks as they fill up the available space. And this is a process called passaging. And you can see on the slide here, you can actually sort of passage usually a sort of larger tissue culture flask from one flask into 4 flasks. So since Alice's SAUS2 cells are considered immortal, and they are an immortal sort of cell line, and are able to replicate consistently, when I started in the lab, I spent a long time culturing and just observing the cells. And during this sort of continual process of culture, I really noticed that the cells are not inert, but are highly responsive to different culture conditions, such as in changes in media. And over the first 6 months or so, I split the original flask of cells 78 times. And I noted that over that time, the cells really started to look quite different from the original cells that I started with. So they became much more sluggish, so slow to move, spindly, which means elongated, and some of the cells even appeared to be multinucleated, so having more than one nucleus. So that is partly why lower passage cells, or cells that have not been split so many times or cultured for long periods, are preferred for research, because they then have less chance of mutation, so phenotype or genotype changes, and are far more in line with the established characteristics of the cells. During this time, it was also fascinating to observe firsthand that cells from a variety of organisms, human and animal, will often really happily coexist in the same flask. And you can see a little GIF animation of some of my co-cultured cells. So you can see they have sort of different shapes. And even during my cell culture experiments, I was even able to successfully co-culture Alice's SARS-2 cells with SF9 insect cells, which is a cell line originally derived from the ovarian tissue of the fall armyworm moth, despite the fact that both cell types usually require really substantially different culturing conditions. So it really shows that close alignment of humans with other organisms. So while the cells themselves fascinated me, knowing the origins of the cells, so the original donors, I was really compelled to think about those organisms, you know, also Alice, and the way in which the total biomass of many of the cell lines now vastly outweigh the bodies from which they originated. So indeed, the first immortal human cell line, the HeLa cell line, which is, you know, very, very well-known cell line, which was established in 1951 from the cervical cancer cells of an African-American woman, Henrietta Lacks, albeit without her, the patient's consent, now have a collected biomass estimated as documented in Rebecca Skloot's book, The Immortal Life of Henrietta Lacks, at 50 million metric tons. So that sort of gives you a bit of an indication how you can go from just sort of a really small number of cells up to 50 million metric tons over a longer sort of span. Now, while SARS-2 cells are less widespread than HeLa cells in a lab environment, the biomass of Alice's cells would also now collectively weigh far, far more than her body ever could. And although the SARS-2 cells were cancerous and therefore mutant cells to begin with, now isolated in the laboratory and continually changing with every passage and also environmental variation, they could also be regarded as becoming increasingly differentiated from Alice's original healthy cells. But at the same time, the SARS-2 cells will always still contain her DNA, and therefore the instructions for creating every part of her body. So cell lines for me really occupy a strange territory, operating in a sense as living and evolving remnants of an often absent or deceased body, and can be regarded as cell and tissue culture art pioneers Oren Katz and Yonat Zur assert as sort of, quote, semi-living, quote, entities, and in many ways a new form of bioengineered life. So in 2008, I translated a lot of these initial observations into the development of several interrelated creative works under the collective title, The Absence of Alice, again referencing the absence of the SARS-2 donor's body. And these really commented on the origins of the cell line, but also foregrounded notions of becoming. So on this slide here, you can see one of the works, which was the wall-based work, which was called 78 Impressions of a Single Object. And this work really reflected on the repetitive process of growing and then splitting Alice's bone cells. Plaster was chosen as the material because it is really commonly associated with mold making, but also the process of duplication. And although the impressions were of a single bone-like object, they were all unique because the resulting shape was determined by the impression process. So the angle of the impression, the pressure that was applied, but also the dryness of the plaster. Over time, as I was doing those impressions, the original bone object also changed because plaster started to adhere to the surface. And in this way, the work really responded to the idea of continual transformation in response to environment and experience. But it was also aiming to highlight the impossibility of gaining an absolute understanding of any object or organism or structure, because our results or our impressions often depend on the particular temporal moment in which the snapshot was taken, as well as the particular methods used. As you can see on the slide as well, the work connected to a range of other companion pieces, such as the multi-panel sculpture Fragments of a Body in the Process of Becoming Other, which was displayed in conjunction with 78 impressions, and also a digital video work in process, which further commented on the continual transformation of Alice's cells. So as I continued my engagement with cells and tissue, I noticed that while some cells such as HeLa cells and SARS-2 cells that are naturally immortalized due to genetic mutations that had rendered them cancer cells, healthy cells can also be transformed into immortal cell lines through the exposure to particular chemicals or the introduction of viral DNA, which disrupts replicative senescence. So essentially it disrupts the stop switch for cell replication. So the cell line HEC293, for example, and you can see an image of those cells on the slide, is an artificially immortalized cell line, which was also established in the 1970s from the tissue of a human fetus. Indeed, the title HEC refers to the cell line's origins as human embryonic kidney cells. The number that often accompanies the cells 293 documents the number of experiments that were required to immortalize the cells, and this was done using adenovirus 5 DNA. So to date, HEC293 cells have spawned at least 12 daughter cell lines, which have been catalogued by researchers in 2018. And this includes the common HEC3T variant, and you can see some of those outlined here. And the 293T variant is very commonly used. And the T signals, again, a further alteration to the cells through the incorporation of the simian virus 40 large T antigen, which again refers to a specific protein from an animal virus. And the addition of that particular protein renders the cell line much easier to transfect. So to introduce additional genetic material, so RNA or DNA into the cell. So that's where you'll find HEC293 T cells are often used for genetic engineering to undergo sort of experiments. So in 2010, I was really fortunate to undertake a residency at Symbiotica, which is a very well-known Australian and world-leading art science lab at the University of Western Australia. And as part of that particular residency program, I worked with HEC293 T cells, but I was able to use plasmids to introduce red and green fluorescent proteins, which are proteins derived from marine invertebrates such as coral and jellyfish, into the HEC cells, which then enabled them to grow red or green under specific light conditions. And here you can actually see just an image of the light microscope image of the HEC cells, and then how the cells were transformed and started to have a glow under those conditions. So as indicated on this slide, plasmids, for those unfamiliar with this, are essentially circular bacterial DNA molecules, and these molecules can replicate independently of chromosomes. So plasmids are used in genetic engineering because they have particular points or restriction sites which have been built in that can be cut with particular enzymes called restriction enzymes. And that enables another gene to be inserted and then sort of stitched back together with DNA ligases. So these plasmids can then be added to microorganisms such as cells using a process called transfection. So the introduction of fluorescent proteins, as many of you might know, is actually a highly established process, and it's used quite routinely in research labs for things such as live cell imaging, studying gene expression, or also observing the spread of disease. And of course, GFP, which is the green fluorescent protein, has been very much popularized in the art world by Eduardo Katz, and particularly his famous transgenic GFP bunny project, in which the artist claimed to have created the first transgenic glowing bunny in 2000. But regardless of the relatively routine use of fluorescent proteins in science and art, I really found it fascinating to observe firsthand the ability to transfer genetic material from different species to facilitate new characteristics. But it also provides recognition of how we, human and also more than human, are in many ways already set up to do this, to share genetic material. And despite being a really routine process, what struck me when working with the HEC293 T cells was how an organism that never really reached full gestation could nonetheless result in a whole series of unique microorganisms with different traits that are in a sense very much primed for further modification, drawing again attention to the increasingly porous nature of taxonomic distinctions. So one of the works that was produced as an outcome from the residency was the work HEC293T, The Transformation of Joni and Oliver. And you can see an image with some detail, installation and also detail shots on the screen here. And the work essentially consisted of a mixed media sculpture and a video work that incorporated tissue culture flasks with the fixed genetically engineered 293 T cells. So those were the cells that had been modified with the fluorescent proteins at Symbiotica. But the work also included taxidermy butterflies and a range of other unsettling elements that merged sort of aspects of the human and also what would traditionally sort of be regarded as non-human, to highlight the interconnections and of cross species and the potential for further transformation, but also to draw attention to the uncanny nature of bioengineered microorganisms that incorporate genetic material from viruses and a range of organisms. So more recently, from 2015, my continued works have really focused on the concept of immortality. So that is a concept that has transferred over from previous work, and also transformation, but here much more in the context of tissue engineering. So in 2015, for example, I was fortunate enough to collaborate with one of the world's leading regenerative medicine specialists, Professor Dietmar Hutmacher, and his team on an art science project titled Biosynthetic Systems. So this project was funded by a Creative Sparks grant from the Brisbane City Council in Queensland, and it aimed to really explore the implications of biofabrication. So biofabrication is essentially the use of additive and 3D printing technologies that are used for regenerative medical applications such as bone and also tissue repair. And on this slide, you can see some additive biofabrication examples from Dietmar's team, including electrospinning, which essentially enables the creation of really fine scaffolds comprised of filaments that are really fine and look like spider silk, and other 3D printing technologies that enable the development of replacement bone structures, but also allow for strength testing for bone scaffolds. So initially, the project was pitched as an investigation into the ethical implications of developing living products, with the plan to create a sculptural bioreactor to showcase a living custom sculpture with a digital representation of its aliveness via a holographic projection. But as with many creative projects, which sort of are much more intuitive, these initial interests really started to shift in response to the input from the larger team and also the research interests. And that, of course, included Dietmar and the artist Bill Hart, as well as a range of experts from the Centre for Regenerative Medicine. And it started to shift to focus more on the concept of immortality through technological intervention. So this interest was spurred by recognition that often popular film and media representations, as you can see on this slide, of tissue engineering often suggests that 3D printed organs and replacement body parts are really close to realization, and that soon we'll not only be able to repair and reshape the body, but eliminate death altogether through the development of bioengineered or robotic bodies, coupled with a form of downloadable consciousness. And being in the centre, we're being with the Centre of Regenerative Medicine experts, that's definitely not the case yet. We've still got a long way to go in the regenerative medicine domain. So the idea of technological immortality is particularly evident also in the views of transhumanists. And I'd just like to clearly state that, you know, this project was very much a critique of these transhumanist views. And transhumanists such as Ray Kurzweil, you know, really are taking a position of transcending the body. But there are also these ideas that are espoused with philosophically aligned projects such as the 2045 Avatar project, which was initiated by Russian entrepreneur Dmitry Itskov. And as you can see on the slide, the 2045 project really proposes the development of a series of really progressive technological avatars, from a robotic body controlled via brain interface to a wholly holographic entity. So there are, of course, a range of concerning issues that accompany this proposition, including access. But really what I'm focusing on here for this talk is the positioning of the body as inherently flawed, and again, technology regarded as superior. So we're looking at that kind of hierarchical viewpoint. And I think that that's really a rather baffling viewpoint when we really start to consider rates of mechanical breakdown and increasingly rapid technological obsolescence that we're witnessing. It also, to me, suggests that, in essence, biology can be reduced to binary code, and thus, again, it sort of has a privileging of mind over body, information over materiality, and it starts to ignore a lot of the myriad interspecies and embodied connections that make up our physical being. So I think it is a bit of a problematic positioning because it is quite reductionist still. So the regenerative medicine team also commented that the regenerative capabilities of the body are still regarded as much more superior to current technological equivalents, and that many of the new clinical products that are currently in development at the centre are actually designed to harness and to support the healing capacities of the body. And on this slide, you can see some of the biodegradable breast scaffolds that are currently being developed for patients who have had cancer. And these scaffolds are actually designed to dissolve as the patient's cells permeate through the structure. So essentially, eventually the structure will be replaced by human tissue, and that then avoids the need for undergoing future surgeries. So through ongoing discussion and experimentation, we were working together, and we used a lot of the technologies that were part of the Centre for Regenerative Medicine's repertoire to develop the project Biosynthetic Systems, which resulted essentially in the development of a whole range of creative works, but also speculative prototypes, which enabled us to test a range of ideas and be a bit more playful rather than fully resolve them straight away. So works that we developed included a sculptural biomonument, and you can see an image here of the work, which featured a Miller hologram of cell growth alongside live 3D printed structures, which were created by mapping the growth and movement of cells on a custom electrospun scaffold. And this work was really developed to test the capabilities of the electrospinning system by integrating text, but it also aimed to really highlight the dynamic nature of the cells. And on the slide, you can sort of see the word imagine that has been spun into a scaffold. And you can see some images of cells growing on the scaffold, which were tagged with green fluorescent protein in order to visualize the growth, which were then translated into 3D printing files, which were printed as part of the exhibition. The exhibition also included a writing machine developed by artist Bill Hart that replicated my handwriting and used a programmed neural network to create an ongoing narrative that reflected on what it might be like to be an AI or an artificial intelligence entity, and also the change from biological to technological. And this work was also presented in conjunction with digital video works based on 3D scans of my face that operated as these kind of strange and fragmented digital avatars. So what we really wanted to do with this work is to prompt viewers to consider, well, what would it be like if you had a fundamental change in materiality in terms of what you're made of, if you're made no longer of, skin and bone, but are actually translated into a really radically different form of embodiment. What impacts might that actually have on consciousness and our experiential capacity? So it was about asking questions. Creative outcomes were also displayed alongside a range of technologies which were developed or used by the Centre of Regenerative Medicine, and that included a 3D printing demo, a whole series of different scaffolds which provided insight into the current developments, but also very much the limitations of regenerative technologies. And here again, we were inviting viewers to consider the potentials of the centre's research, but also very much the philosophical implications of working with living systems, the potential mergers of artificial life and biotechnology, but also starting to signal some of the sort of legal implications around developing these range of technologies for implementation. So as we went on, we saw an opportunity to actually develop a monument and the writing machine further. And the reasoning for that was that we were successful at being included as part of the spare parts program at the Science Gallery in London, which was an exhibition that really focused on body repair and augmentation. And they were really keen to look at those connections with both artificial intelligence and also regenerative medicine. So for the new exhibition, we updated the writing machine, which was retitled Ghostwriter. And in this particular instance, it included an expanded text archive. So the text archive was supplemented also by insight from a range of post-human and also transhuman scholars. And we also included an interactive video avatar and a microphone that then triggered the machine to write in response to detected words and sounds. So here you can see again, I see work as being a continual development. The monument, which you can see here, was also re-envisioned to comment more directly on the human desire to kind of control the elements and nature in order to find the secret to eternal life. So in this instance, the monument incorporated a range of sigils, which were composed of historical and alchemical symbols such as the Philosopher's Stone, as well as references to the transhumanist movement. And it featured a biaxial bioreactor showcasing a range of custom scaffolds in the shape of different body parts. But you can see also on the slide that the monument included videos, which also commented on the power of visual culture and the influence of media, science fiction, and popular science imagery on public perceptions of technological progress. And the pyramid housing at the top of the monument, you can see, featured a kind of holographic video that also referenced the concept of the soul or vital force. So it was really questioning, you know, if we start to become technological rather than biological, how might that also impact on spiritual or religious views? So in combination, the different elements and the two works really were, again, encouraging viewers to contemplate their own position regarding engineered immortality. So we weren't really trying to present a really clear didactic message. So since that work, over the last couple of years, my work has increasingly focused on notions of immortality, but rather here in terms of genetic legacy. So you can sort of, you know, see from this slide that in many ways it does sort of link to, you know, earlier work, particularly with immortal cell lines, but it also is very much the result of my own situation as a single woman in the later stages of reproductive viability. So this really prompted me to consider how I might be able to use biotechnologies to establish an alternative genetic legacy or produce alternative offspring beyond giving birth to human children. And that might include things like engineering organisms, other organisms, to host fragments of my DNA. So this exploration was also inspired by a range of emerging medical processes, such as genetic engineering and gene therapy, which allow the integration of genetic material into host organisms alongside scientific insights in relation to the presence of viral DNA in the human genome. For example, it is now largely accepted that humans contain up to 8% viral DNA, which has been inherited from ancestral infection, and that the integration of viral information might have actually facilitated the transition to placental birth in mammals, illustrating that viruses likely have played a significant role in the evolution of organisms. So drawing on these ideas, I developed the work Self-Portrait 2, Site of Infection, for the 2019 show Pandemic, which was curated by a colleague, Toby Juleff, at the Plimsoll Gallery in Hobart. And this exhibition was actually developed to coincide with the 100-year anniversary of the 2018-2019 global Spanish flu influenza pandemic. But in retrospect, we can see now how the show was also a strange predictor for the 2019-20 outbreak of COVID-19. So the work that you can see here on the slide consisted of a wax cast of my bust. It was very much rendered in a style reminiscent of kind of classical Greek depictions of female deities. And you can see there it emits a vapor which contains my DNA. And by incorporating this sort of DNA breath, the work really alludes to the idea of using viral infection as a potential mechanism of proliferating my genetic material. So essentially, I was speculating that by genetically engineering viruses to include fragments of my genetic code, it might be possible to infect other organisms including humans, and to use the virus as a mechanism to reproduce a bit of my genetic material within a selected host's body. But I also thought that by using a retrovirus and targeting germ cells such as sperm and egg, it might also be possible that the selected DNA sequences could then be passed on to the organism's offspring. And in this way, I could essentially pass on a portion of my genetic information without having children of my own, because other people and their children would then become hosts and continue to perpetuate at least a tiny fragment of me in perpetuity. So I'm sure that this work is making some of you quite uncomfortable. And I just need to say that the work was designed to be deliberately provocative and to really highlight the uncomfortability that many of us experience, particularly post-COVID-19, at the prospect of infection. And the idea of genetic contamination, despite the fact that that in my view, which is a view that follows Timothy Morton's assertion, that we are really not so much individuals, but rather part of a mesh or a multi-species ecology that is comprised of shared and swapped genetic material. So really trying to draw attention again to the fact that we're actually, carry this really long interconnected history and also a history of viral infection in our own DNA. So more recently, my work has returned back to cell culture, but follows the same trajectory, and particularly the prospect of establishing a series of immortalized cell lines, a bit like the hex cell variants, from my own body via a range of processes, such as using viral genes to disrupt programmed senescence, but also to make my own cells available for both scientific and artistic use. So it is worth noting that cell immortalization is now a relatively common practice, and there are even companies that offer an immortalization service as shown on the slide. But in saying this, it's also important to acknowledge that there is absolutely an artistic predecessor, which is the Billy Apple cell line. And this was a project initiated by artist Craig Hilton in 2010. And while the project, The Immortalization of Billy Apples, shares very much the aim to develop an artistic cell line for research across artistic and scientific domains, this project was very much about preserving the artist's brand post-death. So in my case, the desire to produce my own cell line links very much to the previous projects that I've discussed, but it was also developed from my recent experience of uterine pathology. So in 2019, I was diagnosed with a large fibroid, which is essentially a large sort of benign tumorous mass of the uterus, which is really quite common in women of later reproductive age. So I saw this particular health condition as an opportunity to not only isolate and immortalize cells from the fibroid, but also to use the project as a mechanism to consider how creating alternative offspring from the tumorous mass might offer a sense of empowerment and a space for expressing and rethinking the often unspoken grief and anguish of situational childlessness, or also potential infertility, which were things that I was really grappling with during my diagnosis. So prior to having the tumour removed, I developed a series of preliminary works for a solo show titled Morning Story, Expectations, Absences, and the Potentials for Self-Persistence, that really attempted to give some insight into my feelings of grief. And this includes the Memento Mori series, which was a forest of domes representing death, transformation, and hope, and also the life-size sculptural work Coming to Terms with Being Forgotten. So these works, as you can see on the slide, aim to give insight into feelings of mourning for lost possibility, but also a recognition, particularly in Coming to Terms with Being Forgotten, that because we are already connected into a long history of genetic continuous that effectively links us to all organisms and deep time histories, that the idea of establishing a separate genetic legacy is really unnecessary, and that maybe our role is really just to die and form part of a larger cycle that extends beyond ourselves. The work SVKRLM Tumor Baby was also shown as part of the exhibition, provided a contrast, and it shifted from a kind of sense of mourning to a much more sort of hopeful stance of potential, as it alluded to the establishment of a cell line from the fibroid tissue. And by incorporating a flask of fixed cells of mine, which had been established from another project, and taxidermy butterflies as symbols of transformation, you can see that this work makes a direct reference back to the earlier work HEC 293T, the transformation of Joni Oliver, also signalling the potential for the fibroid cells to not only establish a cell line, but also spawn a range of daughter cells that could themselves be open to further becoming. And since this body of work was shown, which was in 2020, my fibroid has been removed, and I did receive ethics clearance from the University of Tasmania in time for collaborators from the School of Medicine to assist with the isolation and cryopreservation, so the freezing of the cells from the fibroid. And on this slide, you can also see images of the fibroid cells, which were successfully grown by project partners, Professor Brad Sutherland and Dr. Jo Marie Courtney at the end of 2020. So the next stage of project development, which has been supported by an Australian Network for Art and Technology grant, is currently underway in collaboration again with the QUT Centre for Regenerative Medicine with Dietmar Hutmacher and the UTAS School of Medicine, and also the Centre for Law and Genetics. And it really involves the cell immortalization, but also genetic analysis alongside other culture techniques that will allow me to extend the use of the cells. So this will include reprogramming the cells into a stem cell-like state using induced pluripotent stem cell technology. And that basically turns differentiated cells, so cells that are skin cells, or other tissue cells such as fibroblasts, into a stem cell, which are then capable of becoming any other cell type. So that means that, you know, skin cells can become brain cells. So this process will not only facilitate the establishment of a cell line and offshoot cells as biological progeny, but will also enable review of procedures involved in making cells available for artistic and scientific research via biomaterials distribution companies such as ATCC. So I'm really looking at, you know, immortalizing and also reprogramming the cells, and then seeing how do you actually get these taken up as a more commercial research product. So furthermore, the development of a cell line and also stem cells will facilitate exploration regarding biological transformation and also cross-species genetic links and difference between original host sort of cells and then the augmented cells. So in many ways, it's really a way for me to rethink the boundary of the body and the self, and to consider alternative notions of kinship, but also the ethical implications of developing genetic offspring that exist as biological products in a commercial research terrain, which is, Oren Katz and Yonat Dur again observe, can really be seen as exploitative of the semi-living. So I'm really interested in how you negotiate that in terms of ethics and values as an artist working in this interdisciplinary domain. So I am sort of coming to time. So to conclude, I really do hope that my presentation has not only given insight into my practice, but also highlighted how art science practices can offer insight into technological developments and prompt reflection on the philosophical implications, and thus reflecting very much a shifting conception of what it means to be human now and into the future. I would just like to say that we are, you know, like our kin, always more than human. And I'll leave it there, but I'd just like to take a moment to acknowledge the many organisations and people that have supported the projects presented today, including this QUT Centre for Regenerative Medicine and also the School of Creative Arts and Media. And I'd like to thank my funding bodies, you know, the Brisbane City Council and also the Australian Network for Art and Technology. So, terima kasih.
Terima kasih, Dr. Serenia Karatz. Very interesting presentation which provokes lots and lots of questions in our head. And one of the questions that have been raised by our audience coming from Swastia. She asked, dear Prof. Serenia, what is the sexiest native germplasm? I don't know what it means. In Indonesia, that is dreamed by Western scientists. Can you answer to this question?
Sorry, could you just repeat that again?
What is the sexiest native germplasm in Indonesia? Maybe this is my limited understanding of the question. Is there such a thing as our native genetic information, and that is seen as the most sensual or most arousing by Western scientists? Is that a thing, or?
I don't know about that. I know that we definitely do have very much a bias towards, you know, looking at genes as still being the be-all and end-all of sort of identity. And that's something that, you know, I'm quite interested in questioning, because, of course, we're learning more and more that, you know, it's not just genetics that make up the human, but we actually have, you know, epigenetic connections, we have environmental connections, we have the gut as well that are really influencing behavior, mood and things like that. So that's also why I'm quite, you know, critical of transhumanist views that really sort of start to think about humans really only existing in genes in their brain. And if we can just harness that, we've got all the information that we need. So I don't know if there really is the sexiest gene that I can think of that come up in Western culture. I think that genetics in general is seen as being still really sexy, even though it's often a very limited viewpoint.
Thank you. Second question from Michael Adi. I quote from his question. I personally consider our finite time in the world as blessing instead of a curse. As we age and eventually die, we aim to build and educate future generation so they are able to create new ideas and new point of view where past generation alluded to. However, if a person can live forever, will the status quo just keep going on forever? Not to mention that socio-economic issues such as those who will be able to afford transhumanism transformation are the 1% or even 0.1%, limiting the birth of new ideas. What is your thought on this issue?
So I think that that's just a wonderful question and observation from Michael Addy. And I absolutely agree. I think that, you know, I'm definitely not a transhumanist. So I position myself very much in the sort of philosophical post-human space rather than a transhuman space, because I do think that transhumanism is very much founded on ideas of human exceptionalism, and are also being espoused by, you know, as Michael has identified, the 0.01% of, you know, very, very wealthy people that don't want to give up their power. So I think that particularly it is a concerning positioning, and it's one that I think is still very much aligned with a Western viewpoint that is about kind of human exceptionalism, power and control, and also not wanting to actually recognize that it is about also giving over for future generations. And I think that that is hopefully a mindset that is starting to shift. And it certainly is a mindset that is very much part of, you know, a transhumanist view, which does look at entanglements. And when you also look at the environmental humanities, and particularly the wonderful work of Deborah Bird-Rose, who was an Australian scholar in the environmental humanities, talked a lot about that need, particularly to draw on Australian Indigenous experiences to be able to recognise the responsibility that we have not only to future generations, but also to other organisms, and that, you know, living our life well, and then passing that responsibility on to future generations is really, as Michael again has indicated, it's really a great honour to be able to do that and to allow for that exchange from generation to the next. So I'm very much in agreement with Michael's position.
Okay. While we are waiting for more questions and responses from the audience, I would like to ask you a question, Serenia. If we are making comparisons between you as an artist and Yossop Prapto in the previous presentation, personally, he changed his environment through his agricultural practices, but also on the reverse, he was changed by the information, the knowledge coming from his practice. Now I want to ask you a personal, if it's okay, personal question. How have you changed after doing all of these explorations, experimentations with human bodies on a cellular level?
So again, I very much do believe that, you know, humans are plastic. So you're constantly changing in relation to your environment. But absolutely, you know, I think that engaging with scientific practices has really made me, you know, be able to almost witness in practice a lot of the interconnections that across species, you know, when you actually see that there's really no difference between, you know, cells from different mammals in many cases, definitely none between, you know, humans from different cultural backgrounds. So it really has given that clear insight, and it sort of reaffirms a lot of the values that I already hold. And I think that, you know, I've been very privileged to be working with a lot of postgraduate students that are working particularly in the ecological domain. And that has been really inspiring for me as well, is to actually become much more attuned and aware of environmental interaction, and also just that recognition that, you know, we really are fundamentally interconnected. You know, I'm really interested in, you know, human waste and things like that. And also thinking about those kind of long-term implication. So it has changed me on many ways, not only in terms of my creative practice, but also, you know, going back to the previous presentation, that when I'm working in my garden, you know, I have a really strong recognition that you don't put pesticides in your garden because it doesn't go away. And I think that you know, if we actually start to really recognize that there is no way, everything is interconnected, there is no, you just do it and it's never seen again. You know, if you put pesticides in your garden, it's going to go into the drinking water. It's going to impact on other organisms in your local environment. And I think that all of those sort of recognitions have certainly played over in how I, you know, work in my garden, you know, how I also clean my house, even, you know, where you go, actually vinegar and water is totally fine for cleaning down surfaces, because I don't want to have an impact on, you know, myself and other organisms more long term.
Yeah. And when you said that we are no different from other types of organism, that's the key takeaway message that I also learned from the eco-criticism literature, that we are less human than we thought we are. But I was wondering, when you told us Alice, the cells of Alice, I keep asking in my head, well, are we still recognizing her as a person, as a subject, through his her cells, or this is just objects, lifeless, although we cannot say it's lifeless, but less than human? What is your insights when you did all these explorations?
I think it's I'm glad you're having that response, because I have the same response. It sort of sits in this really strange positioning where it's both connected intimately to a human, but then it is this sort of small fragment that lives on. So I think, you know, it's something that, you know, Oren and Yonat, who of course are world leaders in tissue culture and art, they've posed that as the semi-living. So it sort of sits in that strange kind of space. And I think that that's probably one of the nicest ways of sort of starting to ask those difficult questions. And unfortunately, I don't think there is a really clear positioning, because it is very much in that kind of liminal terrain. And I think that, you know, although the identity of Alice isn't known, I think that we also have to understand that things are always kind of situated within a social context, particularly for humans. So, you know, the Henrietta Lacks, who, you know, passed away from cervical cancer, but her cells have been used, you know, the use of those cells and so widespread was really difficult for a lot of her family to process when they actually found out. So, you know, I don't think you can really ignore either those connections to a previous body, even though they've since spawned, you know, these kind of bioengineered semi-living entities. So, but at the same time, I think, again, it showcases that, you know, we are this huge ecology of organisms. So in some ways, you know, that also makes me go, you know, I need to eat better for my gut bacteria, and I need to sort of take care of my body, because it is actually also a recognition that you have a responsibility in some ways for the myriad sort of microorganisms that make us up and that allow us to, you know, to think and move and feel and do all of those sort of things. So it sort of shifts, I guess, self-care into a recognition that you're also caring for, you know, other organisms at the same time.
Yeah, thank you, Serenia. Again, I am inviting you to submit your questions through chat box. I also invite Professor Tui Mariantha. If you have any question, feel free to ask your questions to Dr. Serenia. And also, if not mistaken, I also see your collaborator here, Toby, or
yes. So that's very kind that Toby has come along today. So Toby was the curator of the pandemic exhibition.
And yes, we invite questions, responses, thoughts, insights, because obviously this presentation has provoked questions in our mind. I have more answers, but I I don't want to take this opportunity for myself. I want to give more opportunities to audiences. Dr. Tui Mariantha is very interested in learning more your presentation. If there is an article coming out of your works, he will be very pleased to read your articles.
Oh, that's very kind. Thank you.
Yes, and maybe I have one more questions. Again, we have said it again and again about your explorations, probing new terrains and questioning ourselves, and this exploration have implications in our philosophical viewpoint or also ethical understanding of human life. But does it provide any answer?
No, so I think
sorry to ask you this, but
no, and I think that I very much take the position that you know, as an artist, it's not my job to provide answers.
Yes,
my job is really
I agree.
to get you to think about, you know, what is your positioning, and to also encourage people to be more actively involved in, you know, thinking about the, you know, the implications of technology rather than just, you know, accepting them blindly, to actually kind of go, well, what are the impacts that might occur on access, you know, as Michael's question sort of indicates, if these technologies become commonplace, what are the long-term implications? And I think that that's where art, but also science fiction, and I'm a very big science fiction fan, that you could probably tell from some of the examples of the work, to actually engage speculatively in, you know, well, what are the implications if these technologies do start to take place? What are the potential benefits? What are the potential you know, issues that might crop up, and which are the particular trajectories that we are most comfortable with? And that's, for me, particularly around young people, is getting them to actually want to get involved with science and technology, but also with art, so that they're not just accepting technologies that are coming out, but starting to also critically question, you know, how that will impact on their life, and to be able to make better decisions around you know, which aspects we you know, they want to go forward with, you know. And I think that that's particularly an issue around a lot of digital technologies that are coming out. You know, I've just recently got a new iPhone, and the camera, you know, automatically makes you look better. And so I think that that's really concerning, you know, and that's something that I'd like to make some work about in the future, moving into a different terrain, is just how a lot of, you know, technologies that allow for a kind of augmented reality experience, but hidden, how that also changes our expectations, and particularly for young people when they're looking at social media, how that sort of also influences perceptions around, you know, what is normal or not normal, when all of the imagery, or a vast percentage of it, has been digitally altered. So how do we actually give young people the critical tools to be able to navigate these spaces with new technologies? And I think, you know, art, and particularly big art, which engages with asking questions rather than giving answers, prompts you know, the viewer to think about it rather than simply accept that there is an answer. You know, I'm very skeptical about answers, both in the humanities. The humanities, you know, often are also about asking questions rather than giving answers. And even in the sciences, you know, knowledge that has often been accepted gets overhauled when when new developments emerge. You know, they used to say no one gets, and brain cells don't grow. You know, they have a limited number of brain cells. Now they realize that you do have neurogenesis, so you can have growth, cells growing. But it takes that time for a shift in knowledge as well. But we need critical tools.
Yes, thank you. That's a very good way of putting everything together into a kind of summarizing thoughts. I think that art is a way of posing questions about ourselves and also the world, the time of our being as a species, and also our future in the world. We have about 5 minutes, and if you want to use this to make your closing statement, then I think that would be great. Serenia and also Prof. Dr. Tui Mariantha, would you give your closing statement?
Shall I go first, or?
Yes, you go first.
Okay, so first of all, I'd just like to again thank you for the wonderful questions, and also for the invitation to present. I'd also just like to follow on with your final statement and say, that is really what I think the role of arts is, and I do hope that because we are seeing an erosion of creative practice, particularly following COVID. So I'd just like to close by saying, I really hope that we are going to see greater recognition of the value of arts in not only sort of posing questions, but also bringing joy to systems of inquiry. And I think that that's something that I'm certainly motivated by when I do my research, is that I want to do research that brings me joy and that makes me curious. And I think that that is something that I think that that art can really bring, is a kind of joyful critical inquiry. And I think it is very much needed, particularly when there is so much emphasis on getting jobs and on economics, that we actually shift our viewpoint to actually think about who we want to be into the future, and that art can reflect back who we are now, and if we're happy with that, and if we're not happy, that we can make changes towards a better future.
Thank you, Dr. Serenia. And now we would like to invite Professor Dr. Tui Mariantha to give your closing statement.
Thank you, moderator. Actually, I really like the answer of Dr. Karatz, that artists don't have to give answer. This is very liberating. Why? Because in many cases, creative students that's very good in exploring without trapped in theories, they are often asked, what theory you use? And this is very good. Thank you, Dr. Karatz.
Thank you, Professor Dr. Tui Mariantha.
What a frank answer. Thank you.
Thank you. Now we are at the end of our first session, and after this we are going to invite you to join the parallel sessions with diverse presentations, and I want to return the mic to the MC. Thank you very much.
Thank you very much, Bapak Kurniawan Adi Saputro, PhD. And thank you so much, Serenia Karatz and Bapak Martinus Dwi Mariantha, MFA, PhD. Thank you so much. That was a very insightful presentation, and hopefully you enjoyed the presentation. And ladies and gentlemen, this is the end of session 1. Later on, we will have the second session for keynote session, which will be conducted after the parallel session. And we will have a break until 12. So that's why we will have plenty of time to choose the rooms or the parallel session that you would like to join. For your information, we have 3 different rooms for the diverse presentations. We have room 1 and room 2 and also room 3. Each of those rooms offers different topics. So you may choose and scroll around in between rooms. Well, ladies and gentlemen, this is your time to choose the breakout room. You can see those 4 boxes under, what is it? We call it the feature or the menu down there. You can check it. There are 4 boxes and breakout for the breakout rooms, or at least you can see somewhere in your phone, there will be 4 boxes and you can open it and you can choose room 1, 2, or 3. And the parallel session will start at 12 at the midday. So you have time for a break right now. And for the parallel session, you will be guided by 3 different moderators as well. The moderators will be Cahyono Priyanto, PhD, and then Dr. Randa M. Heni Winahyuningsih M. Hum, and also Dr. Raharja, Master of Music. So please take your times for the break and to choose the room. And I would like to extend my gratitude to Kurniawan Adisaputro, PhD, for moderating the keynote session 1, and you will be back later for the session 2. And Dr. Serenia Karatz and Professor Tui Mariantha, thank you so much for the presentation. Well, I guess this is it for session 1. Let's have a break and see you again at 12 in the parallel session. And don't forget to come back to the main room after the parallel session. Thank you.